Good morning, students. My name is Dr. Srinivasarao. So today I'm going to say about the importance of bioinformatics and introduction to bioinformatics. So what is bioinformatics? So this is called as biological information. So what is informatics here? Information means we are going to have the databases. The database is nothing but we have a data. What is the data means? Sample information. So those sample information here, we are going to take the biological samples. So those biological samples we are going to store in your computer. So we are going to store in your computer. So this computer, how are you going to use this computer? So simply we can call as what is that machine learning and softwares. So you're going to use that one for your biological system. So here this bioinformatics is a combination of different subjects we have it. Otherwise we can't work with your molecules. We have a what is the computer science we have it. So you need to link that molecule with the computer science, chemistry, what is the biochemistry, biology, statistics, mathematics. So we need to work together to get a particular databases of your biomolecules. So here, so we'll call as the databases we have it. It's called as biological databases. So these biological databases we have different types of biological databases we have it. So basically, when you have the biology, when I'm going to teach about the biology, so we need to know about the biomolecules. So biomolecule means living molecules. Those are molecules present in our body. So that's called as biomolecules. I can say that. What is that? Proteins, nucleic acids, lipids, vitamins, vitamins and carbohydrates. We can call it these are the biomolecules. But taking consideration of this one, so only we are going to consider the proteins and nucleic acids. Why? Because the proteins and nucleic acids they have a specific sequences. They have their own pattern of sequences. So when you have the proteins, best example I can say that proteins. The proteins are nothing but a building blocks of amino acids. So these amino acids they are arranged in a one pattern. That's called as sequence. So these sequence very important for your protein action and its activity also. So when you have the sequence will change, the sequence of your molecule has been changed down. Suppose we are saying that I have a protein. This protein will have a specific sequence. This sequence will influence your protein folding pattern. The folding in sense we are going to have the expression of your molecular activity. Suppose how it will be act, how it will go into bind substrate. So then how it will go to give the product. So it will also depends. It will all things all depends on your sequence of amino acids. So when you have the change in amino acid sequence, ultimately we will go to have the change in our activity. So I can say the best example of sequence pattern, the sequence of our hemoglobin. So hemoglobin is a composite of what is that? Heme and 2 alpha 2 beta chains of proteins. It's called as proteins. So hemoglobin is a protein molecule. Globular protein we can say that. So these protein molecule will give the structure to your RBC cells. So these RBC cells in concave shape. They are in concave shape because of the presence of your hemoglobin. So this hemoglobin is having the heme plus 2 alpha 2 beta. That's called a globin. So these globulin molecules they have a specific pattern of arrangement. That's called as folding pattern. These folding pattern will have this specific activity shows. It will show the activity how when it binds oxygen and it will supplies to the cells and it will take the carbon dioxide from the cells and gives to the lungs we can say that. It will be outside. So inhalation, exhalation we can go. So this also depends. This hemoglobin. So this is nothing but a protein. So here what I am saying here means your protein. What is that? Your protein is very important. This folding pattern is very important to have the function of your protein. So in your bioinformatics, we are going to store how your protein has been folded. What is the sequence of your protein? We are going to say that one. That's called as protein databases. So we are going to store the information about the protein. So how many times we have in nature, we have different types of proteins, we have it. So those proteins we are going to store it. So those stored proteins we are going to save in our computers. That's called as what is that? We have is that's called a soft copy. We are going to use the different softwares. We can use it. So different languages also we can use it, like your Oracle, Java. We can use it. So those languages, by using these languages, we can do your modelings. We can go for the modeling of your protein. Next one is DNA. That's called as nucleic acids. So in our organisms, so we take example of our cell. our cell is composed of nucleic acids, we have it. So our nucleus, in nucleus we have a chromosome. This chromosome is nothing but so for higher order structure of DNA, we can say that. So your DNA coils, coils, supercoils to form a chromosome. This chromosome will be expressed when you have this instance of new molecule, new cellular system. It will be expressed when you have this instance of new cells. So this chromosome sequence is very important. What is that? The chromosome will be expressed as a form of DNA. So I can say that the DNA is composed, DNA is nothing but a supercoiled DNA is called a chromosome. So this DNA will have a specific sequence. So this sequence is very important. That's called as hereditary, we can say that. So when you change in a sequence, when you change in a DNA sequence, ultimately we will have a defects in our metabolism. Defects on your metabolism. So your genes is responsible. That's called as, suppose take example of DNA. So DNA is nothing but a sequence of nucleotides. So these nucleotides will called as small segment of the nucleotides is called as gene. So big set of your nucleotides is called as DNA. So higher structure is called as chromosome. So here in your bioinformatics, we are going to store the information of your nucleic acids. It may be DNA, it may be RNA, it may be tRNA, mRNA, ribosomal RNA. So those sequences means what you have is nucleotides, what you have it, and in guanine, cytosine, thymine, uracil. Those sequences you won't store it in the form of your computer languages. So you're going to work with that one. So we're going to design your DNA also. We are going to design our DNA. We can go for PCR experiments also, we can do that. So here what I'm going to say about that's the importance of our bioinformatics. So these again we can go for the other one, drug designing. So by using the bioinformatics we can go for the drug designing also. It's called as computer aided drug designing molecules. So this is called as computer aided. This is called as we are going to store all the information in your computer. This is called as bioinformatics. So basically when you work it in your laboratory, we can have two types of labs, we can say that. One is wet laboratory. Wet laboratory means we are going to do with chemicals. We are going to do with our chemicals in your lab. We are going to take HCl, NaCl, water. So we are going to work with that one, like our drug molecules. This is called as wet laboratory. Next one is, I'm going to say that, this is called dry laboratory. Dry laboratory means that's also called as in silico. By using computers, by using your softwares, computer softwares, our mobile softwares, we're going to work with your molecules, especially proteins and DNA. We're going to work with your molecules and you're going to design the new molecule. Those designed molecules you're going to study for the future purposes. This is called as that's the importance of your bioinformatics. So this is introduction to your bioinformatics. So here, so what are you going to do this one? So we first in your bioinformatics, we are going to collect the data. First we are going to collect the data. This is called as data collection. And after collection, we are going to store the data. So because we are going to get this data, that is the databases. So those databases you are going to store in a particular format. That's called as file formats. So that one is again you are going to for analysis. So after analyzing, you are going to discriminate the biological sequences. So here, so these type of bioinformatics databases can be stored in different languages, we can call as. So here basically I can say that, so worldwide many people they are working with bioinformatics. So they have their own institutions, So based on this one, so we can say that biological databases, we can call as biological databases, maintaining institutions we have it. So here I can say that protein databases and nucleic acid databases, protein databases and nucleic acid databases. So here again here protein databases. So suppose here protein database I have it. Again I can say that the protein will possess primary structure, secondary structure, tertiary structure and quaternary structure, because the number of amino acids, what you have it in your number of amino acids. So suppose you have less than 50 amino acids, this is called as primary structure. More than 50 to 100 we can secondary structure. Quaternary beyond that one, etc. We can say that quaternary and tertiary structure. This structure, this count of your amino acids is very important here. So when you want to store your protein sequences, so here I can say the peptide or polypeptide or maybe protein. Maybe enzyme, I can say that. So why? Because so the proteins are nothing but enzymes. All the proteins are not enzymes. Simply I can say that All the proteins are enzymes. All enzymes are not proteins. Some except ribosome is not a protein. It's not a protein, but it shows the enzyme activity. So here, so I'm going to say about the bioinformatics. It will have a database collection. Those database collections, we are going to work it to this one. We are going to design the new drug molecules. We are going to identify what protein sequences. We are going to identify what different diseases in our biological models also. That is use of our bioinformatics. So now databases I can. What are the different types of databases I have? So primary databases and secondary databases, tertiary databases. Like this we have it. So basically I can go with the protein sequence databases. The protein sequence databases again divided into two types. One is protein sequence databases and protein structural databases. Again nucleic acid. So nucleic acid sequence databases and nucleic acid structural databases. So here the sequence will influence your structure. So first we are going to study the sequence databases, protein sequence databases. So here these protein sequence databases can be maintained by different institutions. Suppose the protein I have it. So our body is composed of all the proteins, more than 6,000 proteins we have it. But storing this information, the protein information is very, very tough because I can't work it. So best example I can say that, so we in our body we have more than 600 proteins we have it. So we need to classify that one, we need to what is the sequence, what is the target, what is the source of the molecules, how it will produce, what is the gene expression levels. So everything we need to study that one, we need to give the information. Otherwise it may be, it's a waste of our work. So here, the protein information databases again divide into what is that? Three types you can have it. Primary sequence databases. Then next one is composite databases and secondary databases, secondary sequence databases. So primary sequence databases means they are the institutions. So many institutions we have it. Those institutions, they maintain the primary sequences of our protein. So they don't go for other protein, other molecules. They don't go with other molecules. Okay, simply I can say that suppose we have a Nagarjuna University. So these Nagarjuna University we have a fund for the research. So my university will self only maintain the primary sequences. It is not possible to maintain the whole information because strength of the people and the working load. So it won't be we can't do that one. So here each and every institution they have their own capability to do the research. So primary sequence databases. So here we have a peer institution, Swissport, the different types of institutions we have it. Those institutions they will maintain the only primary sequence of our protein. The next one is composite sequence databases. So here these composite sequence databases means what do you say that 2 or 3 primary institutions. Suppose peer I said you, protein information research, PDB protein databases. There are institutions we can call as the combinedly. To 1 or 2 institutions, 3 or maybe 1 or 3 maybe, they combinedly do other work also. They do the other related database work also. That's called as composite databases. So myself, suppose other than Acharya Nagarjuna University and Andhra University, these two we have it. Then we have a SKD, Sri Krishna Deva University. So the other